From the idea to the finished pharmaceutical product, it is a long way throughout the whole organization with many obstacles. Data fragmentation leading to separate data silos, cumbersome processes, slowing down value-add creation, lack of knowledge how to capture and share important information between the departments involved. It all costs time. But in the development of pharmaceutical products, every second counts. Let's make them count. Digitalization creates speed. As always, communication is key. The key to a fast and efficient production. Enterprise recipe management helps you communicate digitally via software to directly integrate formulation, laboratory testing, and recipe designers into the production world. Thanks to an easy, scalable digital transformation enabled by Siemens Xcelerator, our software closes the gap between R&D and production, integrates all process data for machine learning in real time, creates a digital backbone for knowledge and data exchange, simplifies the scalability of recipes even for worldwide production, generates a production digital twin for virtual visibility of the future real world to hurdle unforeseeable problems, and most importantly, it helps you win the race against time.
Thank you, everyone. It's a pleasure to be here. Welcome on this digital journey. It's an exciting time. We're going to show you how we can move forward with end-to-end digitalization. So let's first talk about what are the vision statements and goals of enterprise recipe management. So we first have accelerate new product introductions. So that's really the key, is how can we help our customers digitize the end-to-end with really the business goal of getting new product introductions through the organization faster, better, cheaper, with a data-driven knowledge-sharing solution and a scalable end-to-end recipe management solution. So the high-level strategy consists of a backbone for knowledge model orchestration and workflow management. So you'll see on the top the drug development lifecycle, the backbone, and then we have various microservice applications that can connect into this data hub into the backbone. These microservice applications, as you can see, are color-coded. This is representing that this is a vendor-agnostic digital approach to help become fully digital end-to-end. Some applications will be delivered by Siemens and other applications by non-Siemens organizations. But the whole goal is that the connection of these functions, these applications working together, connected into a digital hub, allows you to create this digital backbone and create products to go through the product lifecycle much faster. So now we're going to take a deeper dive into the detailed strategy. So this is going to be a little bit complex, maybe challenging for some, but I think it really does tell the story. So we first have on the top, again, you will see the drug development lifecycle. We have a process PLM layer, which is the backbone, which is what you saw previous. Inside of the backbone, we have several swim lanes, several functional layers. These functional layers consist of a data hub for connectivity, our PLM layer, which is our product lifecycle management layer for overall orchestration, a knowledge graphs to help take data across the organization, run it through knowledge graphs to really contextualize the data, because it's all about contextualizing all of the data so that you can make decisions on this data. And then a user cockpit so that you can enter into the system based on function and user persona. So now let's start the journey. So how does this work? We go through our digital twin tools. So we have various simulation tools to both simulate the product and the process. And then we open up a project in our PLM workflow. After the project, we define our QTPP requirements. This is the set of product requirements that we want to make. So quality target product profile. The QTPP requirements then get passed down to our product and process development space. In this area, we will be doing several functions. We will be doing our planning, our early process definition, but also, more importantly, our design of experiments. So this one tool has both an early process definition and design of experiments all built into one tool. We execute those experiments. We analyze the data, and we come up and we create our first PD recipe. What's important about this PD recipe is the first electronic version of a recipe, but yet still in the R&D labs. And this is a very unique tool and a very unique approach that in order to digitize the end-to-end, you have to first digitize the lab. And by developing this early PD recipe, you are starting that digitalization process. You will see inside of the PD recipe, you have several elements. You have critical process parameters, which are your set points, critical quality attributes, which are your quality measures out of the system. You have your formula, your bill of process, and your bill of material. These elements are defined early in the R&D lab as opposed to further downstream. By developing these early in the lab, now we've become digital, and now we can pass these digital elements through the rest of the life cycle. We also will be connecting to, say, existing ELN systems, electronic lab notebook systems, LIMS systems for testing, and also PAT systems, so process analytical technology, really designed for multivariate analysis. And then as you can see below that is the instrument data orchestration layer. So we also will be connecting electronically to lab equipment, smart lab equipment. Could be HPLCs, it could be liquid handlers, it could be anything that's electronic that can send us an electronic signal from the lab directly into this product and process development functional area. And this is the key, again, to gather that data, contextualize it against a process. After we've iterated through and met our target goals for quality, we then promote it to what is called a CMC module. So the CMC module stands for Chemistry, Manufacturing, and Controls. So this is the CMC module. Think of it as the data concentrator for the R&D labs, where you're gathering all the data, you're taking this data in a specific format, and then you're submitting it to the regulatory authorities to be able to create your clinical trial batches and release those clinical trial batches to the market. So this is your first approval, early approval of being able to introduce clinical trial batches. The next step of the journey is as you're going through different lab scale-ups inside of the lab, you're going to be doing small scale lab, medium scale lab, large scale lab, maybe even pilot batches, still R&D lab based. You're going to iterate until you hit those quality targets. Once you hit those quality targets, you're going to then promote to what is called a general recipe. The general recipe is now the starting point of ISA 88, which is the industry standard for batch and recipe execution. So now, as you can see, now that we're inside of a general recipe consisting of process actions, we're then going to promote down the workflow to a site capability check. So the site capability check is a rules engine with an AI backend that allows the querying of production equipment so that you can best decide where am I going to produce this set of process actions of my general recipe. So this could be a querying of all of your production areas globally. It could be also a contract manufacturer. But wherever the best applicable automation and equipment that I can merge with this G recipe is what this verification, this site capability check provides. Once we find our best candidate to produce this general recipe, we then merge that general recipe and tech transfer it to the site recipe. So the mechanism here again is merging the process actions of your G recipe to the equipment capabilities. The merging of those two is what creates your site recipe. So now that I'm in my site recipe, I want to localize that site recipe to the specific quantity with some more specifics, and that's going to generate the master recipe. I then take that master recipe, I merge it with a work order coming down from your ERP system, could be SAP, and then the merging of that with the master recipe creates the control recipe. The control recipe is then released on the MES and batch and automation layer systems. So this is now released to actually create the product. So by working through the MES set of manual instructions and automated instructions controlling your batch engine and PCS7, now we can go back and actually create that product, release the product to the market, and then finally pull in post-market surveillance of the patients and the market back into product and process development. This is also meeting the needs of the new standards coming out, the new ICH guidelines, requiring feedback loops, post-market surveillance. The area that you see in yellow is what is considered digital labs. So we have many tools in digital labs that are working together to really make this digital journey, to make it happen. I mean, you cannot just digitize manufacturing and then not think about the early labs, because you cannot transfer Excel and paper into basically the manufacturing systems. So you first have to digitize the lab, and then you can enable this entire end-to-end flow. And then we're going to highlight one specific new product that we've acquired, re-architected. Now it's available on AWS, our Siemens AWS Cloud, as a SaaS offering called RIFNX. So what is RIFNX? This will just be a quick overview of this tool. So RIFNX is really a very unique tool designed for R&D labs. RIFNX provides the ability for the R&D scientists, instead of describing their process steps when they're doing their lab, let's call it their small-scale lab runs, instead of documenting in an ELN or Excel or paper, they're going to actually create an early process definition in this process designer space. Very simple to drag and drop. You're creating a mini workflow of your steps in the lab. Then below that, you'll see experiment design. This is where you're defining your design of experiments, or DOE. This is what's called a hypothesis model tool. Hypothesis model means that I have my early hypothesis, which is my process definition in my process design in the flow blocks. And then I'm going to run experiments against my process definition. And I'm either going to prove or disprove that process definition, and that is the hypothesis basis of the model. Now I'm going to execute the experiments. The experiments, you can run 10 experiments, 100 experiments, or 1,000 experiments, but you're running all of these experiments, but what are we really trying to accomplish? If you think about making a new product, you have a huge multivariate design space. You're trying to shrink the design space down to the optimal set of conditions to make this product. And this is what's occurring with this experiment design against process, capturing the data, and then analyzing the data through various analytical tools. And you can see the analytical tools. We most often use JMP, but you can use any of the tools listed above and more. To add to this tool has roughly 200 APIs to connect to multiple systems so that it can be part of your ecosystem. Last slide. Just a couple of customer benefits of use cases of using these tools and also becoming digital. So Novozymes is the leader making enzymes for the world, and they have greatly reduced their time to market, their new product introductions, by digitizing the lab and then moving that digital into the pilot and then into full-scale manufacturing. And then you can see the other types of value benefits. And I'm out of time. So, as most of you know, I could continue talking about this for a long, long time. So please come over to the booth and we can dive deeper and then talk about how applicable it is to you. Max?
Thank you, Todd. Thank you. And yeah, I think you mentioned it before. That slide was complex. It was a complex diagram, but I think you did an amazing job of guiding us through nice and slow, nice and easy. And yes, if there's any further questions about that diagram or anything else you've heard about these digital labs, then feel free to come and speak with Todd. Thanks again,
Okay.
and enjoy the rest of ACHEMA. Thank you.
Thank you.